Now, it is my great pleasure to introduce our next speaker, who is our keynote also. He is Professor Manolis Kellis from the Computer Science and Artificial Intelligence Department, a computer science course. He runs the Kellis Lab at the MIT Computer Science and the Broad Institute. He is a true dyed-in-the-wool MIT not creation, perhaps part of our very DNA, if you want, or part of our very structure here. He transcends field boundaries like you wouldn't believe, and does with computers what biologists could only dream of, and oftentimes teaches us things about biology that come from computer science instead of biology proper. The amount of work that he's put out that is actually truly impactful is amazing. Let me just list a couple of things from single-cell genomics, which I think you'll talk to about today, variations in disease, epigenetics, which is a huge, wonderful opportunity right now. We've discussed this with him earlier, and we are both in agreement that olfaction is at the forefront of these things. You can change your olfactory perception by just thinking. You can change your epigenetics by just meditating. And somewhere, the genomics and genetics and epigenetics of all these things connect to olfaction, connect to neuroscience, connect to health and disease, connect to appetite, metabolism, and obesity. So I give you Manolis Kellis, one of the most sort of MIT people you will meet even inside of MIT, and also I believe the youngest ever tenured professor. I think
Maybe not ever, but pretty close.
Well, definitely one of the youngest tenured professors.
How can I plug in?
Please, welcome our speaker.
Thank you. Yes, plugging in would be great.
Okay, you're good.
And then
What do you need?
projector.
What do you have here? This will work. To the
Yes.
Okay, good to know. And relax. You're penultimate. Do whatever you want.
That's awesome. So I am excited to see everyone at the Museum of Science. I don't know if you've told people that we're heading there.
Right, maybe you should announce it that at the end of this, after Shugwang's closing remarks, we will meet at the Museum of Science probably around 6 p.m. or so. We'll give you the exact time. And it will be a semi-guided tour of the nose exhibits there, as well as any other exhibits. Everybody's on their own getting there and on their own getting their tickets. But we will guide you and we will have fun with you. And the museum closes at 9. We should be there at 6. And after that, everybody can go to dinner.
And I'm bringing the kids.
Yes, that's a wonderful
I just realized that my background is always my kids. I can just do this to. So they'll be there, and they hope to see all of you there. And I'm just thrilled to be here. So basically, Andreas stopped by my. Hold on 1 second. I have a phone somewhere.
Whoa, look at it.
He knows to normally do.
I thought Manolis was the goat as a time, but he's actually a goat.
Yes, exactly. Speaking of goats, I was actually kayaking in Greece to this island called, anyway, on the way to Sunion. That's basically about a few kilometers from shore. And as I'm arriving in the midst of these giant waves, I'm basically seeing this completely vertical block of rock and these black dots on it. They were not trees, they were goats. They were just like at the edge of the cliff. So I'm trying to take a picture of them as the waves are pounding on me, but I don't have quite a clear picture, but that sensation of, wait a minute, these are animals. So anyway, my mom always jokes that she was a goat in a previous lifetime, because every time she goes on a mountain, she's just like on the cliff. So anyway, I feel like I'm here among family. I think Andreas just is the epitome of the sort of next generation scientist of the Renaissance. I feel that every time I speak with him, my brain expands and I'm learning so much more. So he messaged me the other day and he was walking by Stadas. I'm like, hey, do you want to jump on Zoom? He's like, I'm actually downstairs. Where should I find you? And my schedule was packed, his schedule was packed, and somehow we carved out an hour and a half of just brainstorming about olfaction and the brain. And
helped organize our meeting.
so it's really a true pleasure to be here. Thank you for putting together such an amazing program, because I think that, as we heard earlier, this is like the last remaining sense that we need to understand. So I'm going to tell you a little bit about sort of what I do in my day job, which is sort of try to understand disease circuitry. And then I have a few thoughts that I'd love to share just from our sort of short brainstorming session on sort of the connections between deep learning, olfaction, and more generally sort of the future of perception engineering, if you wish. But let me tell you what I do in my day job. So basically what we're trying to do is understand the molecular basis of human disease, understand the mechanism of human disease. So throughout humanity's history, epidemiology has always been about correlation. You know, so to give you an example, the people who can drink Starbucks every day have better health outcomes. Is it because Starbucks coffee is so healthy for you? Or is it because the people who can afford Starbucks every day can also afford better health, better nutrition, better exercise, more time for leisure, better sleep patterns? Certainly not in this crowd. But everything's correlated. And figuring out causation requires two things: either clear temporality or perturbation. And the one place where we can actually get causation out of correlation is actually human genetics. If I inherit a genetic variant and that variant correlates with an outcome, it's very unlikely that the outcome is actually causing that inherited variant that is running through my family. So basically, genetic epidemiology allows us to get a causation. And that's sort of the basis of the open door that we have to understand mechanism. Because everything else is correlation. The one place where we get causation is if we can find genetic differences between individuals that correlate with a phenotypic outcome, and then we figure out the mechanism through which they work, and then we go and perturb that mechanism. That's when we have a dramatically improved chance of actually having a therapeutic that works. Because all of these studies that have basically looked at curing mice from every ailment you can imagine, we have the world's healthiest mice, you could possibly believe in science. Every single time we try to take that from mouse to humans, there's some kind of back door and pathway where the human system just reacts adversely and sort of, shuts that door. But the moment you have human genetics, then you have a path to actually perturbing that mechanism. So that's what we do in my lab. We basically try to sort of use that window of opportunity through genetics to actually understand mechanism and how to reverse that mechanism. So basically how to enable therapeutics from genomics by both dissecting, understanding, and manipulating disease circuitry at single cell resolution. So that was my talk. Thank you very much. That was great. All right. So what is genetics? Genetics, we've known about genetics forever. Basically, for thousands of years, humans have selectively bred crops to basically make them edible, animals to actually make them, huggable, and wool to make it wearable. So we've also understood inheritance in humans. I have to say the joke here. Basically, when my blonde, blue-eyed, pale son was born, everybody was like, "Oh, where does he get his blue eyes?" And I would always say, "Well, his dad, probably." And of course, my wife eventually sent me the 23andMe comparison of me and my son, and she's like, "You can stop making that joke now." But for a geneticist, the answer of where does he get his blue eyes or blonde hair is obviously both parents, because it's a recessive trait. And that's something that we actually had not understood, the fact that you can have brown-eyed parents for dozens of generations, and then a blue-eyed kid. You don't need to kill your wife. This is just a recessive trait. The first person to truly demonstrate the mechanism through which this happens is Gregor Mendel, through his experiments in peas, by basically showing that dominant and recessive traits can basically lead to this independent assortment, these discrete units of inheritance, these dominant and recessive alleles, and the lack of blending. This was genetics completely worked out in 1866. Gregor Mendel sent his manuscript to Darwin, and 50 years later, when Darwin had died, we found a manuscript on his desk that was still unopened, and it was Gregor Mendel's manuscript. His paper was cited 3 times in 50 years. no one believed that the laws that Mendel had so eloquently dissected in peas actually applied to humans. And the reason is that humans understood about continuous variation, about the fact that if you look at hair color, it's not just like one of three options. It's a continuum. Eye color, it's a continuum. If you look at human height, it's a continuum. So how do you reconcile these continuous phenotypes observed in humans with these completely discrete 1-0 genetic inheritance of Mendel? And that was not done until 50 years after Mendel's discoveries, with Ronald Fisher and other statisticians basically coming in and saying, hey, even 5 Mendelian loci would basically give rise to a semblance of continuous variation. And that's extremely key to realize, that basically it seems continuous, but it could just simply be additive effects of a small number of traits. And in fact, just a week ago, the top of my Google Newsfeed, again, it's an adaptive algorithm, so it's maybe my Google Newsfeed, is about sort of this comprehensive mapping of human height variation. There are now thousands of loci, every one of which contributes reliably to your height at the scale and magnitude of less than a millimeter. So what am I saying here? I'm basically on one hand telling you, we have causality, we can manipulate the system. On the other hand, I'm telling you that it's not just 5 loci, it's thousands of loci, and not just for human height, but for every single aspect of human biology, of human cognition, of human metabolism, of human olfactory perception. Every aspect of human biology is minutely nudged by thousands of genetic variables, and of course, as you all know, by huge environmental components. So why am I so focused on identifying the genetic circuits underlying these loci? The reason is that when you identify these loci, you have a path to manipulation, and you're not constrained by the minute changes that these genetic variants carry out. You can actually find these circuits and manipulate them a lot more. So basically, just to give you an example, we took the strongest genetic association underlying obesity, and we narrowed it down to a single letter, a T to C alteration. Basically, if you are homozygous for lean predisposition, you have this AT-rich motif. So the vast majority of humans have this AT-rich motif that is bound by this AT-rich interacting domain factor. Okay? The genetic variant is very unusual in that it actually has an impact of 7 pounds in an adult. How can a single nucleotide have an impact of 7 pounds in an adult? It is because it actually rose from 5% frequency to 45% frequency from Africa, in Southeast Asia, in Europe, and so on and so forth. So 42% of European chromosomes actually carry the risk allele. I'm actually homozygous risk for the risk allele. I'm constantly starving and constantly exercising in order to not be obese, because my whole family has that predisposition, and I got 2 alleles from both mom and dad with the risk allele. So what does that do? So basically, if you don't have this AT-rich motif, which is normally bound by ARID5B, that then represses this massive switch. It's not just a little switch on the wall. This is the control panel that we have in the basement for the whole building. So that 12,000 nucleotide enhancer, it's like enormous. Most enhancers are 500 nucleotides. This is 12,000. This 12,000 enhancer is basically at the root of a developmental program that decides if my cells are going to basically store energy as lipids, this is white fat cells, or if they're going to burn energy as heat. And basically this decision is made in the first 3 days of differentiation. Every single time that my fat cells renew, they basically decide if they're going to be fat storing or fat burning cells. And evolution doesn't allow big variations in general. So basically it's very rare that such a strong effect variant rises to high frequency? The reason is probably that in the Out of Africa event, when we had to cross deserts and ice ages, etc., humans had to somehow store energy more efficiently. And if you look actually retrospectively, you can actually do retrospective genetics with the Framingham Heart Study that has been basically following individuals across families for multiple generations. You can actually go back to the blood samples from before World War II and look at the correlation between this genetic locus and obesity. And you see no correlation. And after World War II, huge correlation. What happened? Office jobs, McDonald's, fast food, dramatic changes in the environment, and those of us who were doing great and were well suited for storing calories through ice ages are suddenly faced with an abundance of free food and little exercise. So suddenly, that variant that was strong effect and therefore would have been kept at low effect by evolution was tolerated to rise to high enough frequency. And as the environment changed, it became a deleterious variant instead of an advantageous variant. So we are now referring to this as an obesity risk allele, where the risk basically increases your chance of a disease. But in general, we should be thinking of not risk and non-risk because it's all environment dependent. So for example, the sickle cell risk allele is the allele that will keep you alive in Africa in presence of Anopheles and malaria. Basically, the geographic distribution of the sickle cell allele matches exactly the geographic distribution of where malaria is prevalent. And in that environment, it's actually extremely advantageous to be heterozygous for the sickle cell allele because it actually protects your blood from actually carrying out the pathogen. So in the context of this relatively weak effect of 7 pounds, I mean, I can't tell my family, hey, I'm obese because I have 7 extra pounds, but what I can say is that now that I understand the circuitry, I can actually perhaps manipulate that circuitry. And by uncovering this circuitry in this incredible team, I like to joke that we wrote a paper about 1 bit of information in the human genome. So basically, this is the New England Journal of Medicine. As you know, it has an impact factor of something like 80. And we basically wrote about a single letter alteration. But what we did is actually figure out how that letter actually works. And that letter basically is bound, results in this. And I was introduced at a talk by the head of one of the big pharmas in this annual meeting bringing together academia and industry as the author of the most depressing paper of the year. And the reason is that if you look at this genetic association, it basically sits squarely on the FTO gene. There's no doubt that there's only one gene underlying this genetic association. So all of pharma was like thrilled. It's like finally we know the target, because for many of these genetic associations, you have 5 or 6 or 10 genes sitting under this genetic region of association. And in this particular case, there was only one, so people were so excited, and they invested more than $1 billion collectively across the community on understanding the function of this gene. And here we go, and basically infer a circuit in our paper titled FTO obesity variant, where FTO doesn't even appear in the circuitry. What's going on here? What's going on is that this sits right in the middle of the first intron, the non-coding portion. So if you look at the scale here, it's quite daunting for those of you who are not spending your days looking at genomes. Exon 3 is this tiny little dot. Exon 2 is this tiny little dot. Exon 1 is, beyond the edge of the slide. So 1.5% of the human genome codes for protein. We basically have 100, centimeters, and one of them is going to be, the exons, sprinkled among very large non-coding regions. And in this particular case, the genetic association basically spans 89 common variants. Just to give you a little bit of background, so we might look like a population of 6 billion people. We're an effective population size of 10,000 people. Basically, human genetic variation was fine and well in Africa, and that's where most of genetic variation still lies, but a very small group of people went off and then populated the rest of the planet. So if you look at our effective population size, which is a measure of the diversity of humanity, basically how many mutations have we accumulated, that dramatic population growth doesn't count. We are basically still a very small population. And if you look at the number of independent genetic alleles, independent blocks, then what you see is that from mother to son and from mother to daughter and from father to son and father to daughter, you basically have blocks of co-inheritance. So you don't just inherit that one nucleotide, you inherit a whole block of 50,000 nucleotides with all of the variation that sits within that block. Everybody with me here? Mm-hmm. So there are 89 common variants that are co-inherited in this locus, only one of which is in fact the driver, as we showed here. And to go back to why this story is so depressing, that variant that basically sits in the middle of this gene, in fact, doesn't control this gene at all. It controls two different genes that are sitting 1.2 million nucleotides away. So at the scale of this picture, this is basically beyond Harvard. This is, I mean, this is extremely, extremely far away. So that basically means we can't just look under the lamppost. And that also means that we need to understand the circuitry of the human genome. We need to understand in every region of genetic association, what are the nucleotides that are co-inherited, that are associated with that disease? What are the regulatory control regions where these nucleotides are found? What are the regulatory motifs that these nucleotides disrupt? What are the regulators that bind these letters, these nucleotides? What are the tissues and cell types where these control regions are active? And ultimately, what are the target genes through which the biological effect happens? Is everybody with me here? Awesome. So that's what we need to do for every one of 120,000 genetic associations. Okay? So basically, in the time of Mendel, he had like a few dozen traits, and that was nice and good, and they were all independent, except for a couple where he had to possibly fudge the data to actually make them look independent. But what Morgan and Sturtevant actually discovered is that the slight, quote-unquote, inaccuracies, the slight deviations from the law of independent assortment that Gregor Mendel had discovered or had inferred from his data, these slight deviations were in fact the basis for genetic mapping. Because if two genes, which we now call genes, are segregating somewhat dependently, then that means that they're close to each other on the chromosome. And that's how dozens of Mendelian loci were mapped in the 80s, by basically tiling the genome with markers and then looking in large families of who inherited which version of a trait, and then mapping the trait inheritance pattern with the marker inheritance pattern, thereby leading to that colocalization and ultimately finding the genes for a bunch of traits. With complex traits that basically by their nature have thousands of underlying loci, this is not possible because you cannot track segregation of 1 millimeter differences in height. We vary more between the time we wake up in the morning and the time we go to bed in our height than any one of these thousands of statistically reliably variants allow us to recognize. So how do we deal with all that? We deal with all this by basically understanding that circuitry that I mentioned and then being able to now go in and manipulate things in a way that's much more brutal, quote unquote, than evolution would ever tolerate. So evolution is constantly acting on the human genome. I mean, it is ridiculous to think that somehow humans have escaped evolution. It is constantly acting at every stage of sperm, ovary, zygote, development, There's all of these stages. Why do you not tell your friends that you're pregnant until you're 3 months old? The reason is that there are many embryos that simply don't develop after 3 months. There's massive amounts of checks that constantly happen to basically give rise to the amazingly perfect individuals that I see in this room. I mean, I'm serious. Like, every single one of you is a huge success story of alleles and gametes coming from the parents, and then starting from one cell, leading to thousands of cell divisions, to trillions of cells, with 3 billion letters of DNA functioning amazingly well. It is remarkable. So I want you to maybe call tonight mom and dad and say thank you for making this miracle of life possible for each of us. And the genetic differences that are common in the population have these minute effects. But if you look at what we can do therapeutically, we can basically go beyond what evolution tolerates in the population. And we can actually start manipulating these at much stronger effect. Okay? So the analogy that I like to give is that the volume that, I don't know, your neighbor likes to tolerate is very low. But then the moment you go in the lab, you can just crank it up all the way one way or another way. So basically evolution only tolerates turning these knobs a minute amount. But once we figure out the knobs, we can therapeutically go in and change them a lot more. And that's what we did. We basically said, okay, great. Let's first of all show causality. Let's show that even changing that one letter can actually lead to this dramatic change in cellular phenotype. And indeed, so my cells are unable to thermogenize. They're unable to burn calories as heat, to heat generate. But if you take cells from, basically someone with my genotype, and you CRISPR-Cas9 genome edit the C, which disrupts the AT-rich motif, back into a T that restores the AT-rich motif, then suddenly you're able to restore thermogenesis. You're able to sort of make thermogenesis work again. If you go into mice and you basically say, let's increase or decrease the expression of these downstream target genes that are a million nucleotides away, what you basically see is that the mice lose 50% of their body weight. Because now it's no longer a minute effect. Now you're going in therapeutically and you can do much larger effects. And when you put the mice on a high-fat diet, the mice that are sort of normal genotype gain weight, as anyone would. These mice where this is stuck in the thermogenic position are just burning the calories away when they're awake and when they're sleeping. They're just not changing their appetite. They're not changing their exercise. They're just simply burning calories. So for those of you who want some of those pills, I have them right outside in the back. Just come talk to me. So that's what we want to do for every single one of hundreds of thousands of knobs that we have now found. Everybody with me? So what makes it hard? What makes it hard is that in 93% of cases, these genetic variants are not perturbing genes. They're not perturbing the proteins. What they're perturbing is these sprinkled light switches that are scattered across the human genome. So basically, these hundreds of thousands of genetic associations are extremely difficult to interpret. Okay? So basically, the promise is, woohoo, we have a genetic association. It sits in FTO. Great, let's spend $1 billion. We'll figure it out. Well, we need a path to causality. We need a mechanism. We'll find new target genes, new therapeutics, enable personalized medicine. The challenge is that in 93% of cases, you have this picture, where you have a bunch of variants, not a single one. The target gene is not known. In this case, it was a million nucleotides away, several genes over. The causal variant among all of them is not known. The cell type of action is not known. The relevant pathways are not known, and therefore the mechanism is not known. So you can't intervene. You're stuck. And that's what we're trying to change. How do we change that? By basically understanding how the genetics ultimately acts through every aspect of human biology. So what we need is basically a map of every aspect of human biology. Simple. So we need every cell type across thousands of human cell types, across every individual, across billions of individuals, across every developmental stage, across every disease state, across every perturbation. right? Yes, that's actually what we do. There's no shortcut. Well, except for the fact that if we understand these common variants systematically, then we can bring in machine learning and deep learning and algorithms to understand the rules of how the genome actually works, and then predict that circuitry. Okay? So what we're doing in my lab, despite being a computational lab, we basically burn about $20 million of NIH money every year profiling the heck out of huge cohorts for brain, for heart, for metabolism, for every aspect of human biology that we can get our hands on. Okay? So what we're looking at is human samples from open heart surgery, from epileptic surgery, from muscle biopsies and adipose biopsies, post-mortem samples across every region of the human brain, across thousands of samples, to understand how the RNA changes, that's the expression of the genes that everybody's focused on, and how the epigenome changes. What is the epigenome? The epigenome is how DNA is wrapped around these nucleosomes. We talked about these 147 base pairs, which corresponds to 7 turns for each loop of this DNA helix, the most beautiful, noble molecule of our time, wrapped around these nucleosomes, and how these nucleosomes at roughly 200 nucleotide resolution are basically packaged up the way that you label your boxes when you move. When you move, you basically write living room, kitchen, kid bedroom 1, kid bedroom 2, etc. And when it's time, when you're finally in kid bedroom 2, you open up the kid bedroom 2 boxes. That's exactly how the epigenome works. So you basically have colors, post-translational modifications in the tails of histone proteins that make up nucleosomes, colors, like Post-it notes, that basically say, if you're a liver, open this box. If you're a neuron, open that box. Everybody with me here? So that's what makes a single, talk about an amazing machine. We have a single code, a single automaton, which then self-replicates and then sends signals to each of these cells to differentiate, to package up the stuff that won't be needed, and to open up only the liver regions based, and then all of these form these networks of interconnected neurons, olfactory perception, biological immune functions, and every aspect of human biology, physiology, cognition, etc., is basically enabled by the diversity of collaborating cell types, even though they all magically, I have to say, share the same underlying code. So that's the epigenome. The epigenome is basically this master integrator that basically brings in information from genetics, of course, from disease, of course, but also from the environment, about socioeconomic, chemical, pollutant, age, sex, comorbidities, and of course, all kinds of biological covariates. So basically, this epigenome is this great integrator that we can use to now start understanding how every cell turns that common code into a biological function. Who's with me so far? Yes? Awesome. Who feels that they're learning stuff? Yes? Awesome. Great. I do that in my lecture too, and it's so nice to see thumbs up. This is great. So basically, what we have done in my research group is understood the language of the epigenome. We've basically written machine learning models for systematically understanding what language does the epigenome speak. How does it encode different types of functions? We've discovered distinct signatures for proximal regulatory regions known as promoters, distal regulatory regions known as enhancers, active transcribed regions, repressed regions with a bunch of different modifications. And we've basically done that systematically across not only different tissues of the body, but also different individuals that are all healthy but differ genetically, or I should say and disease individuals that also are modulated by these genetic variants, but also have the disease component perturbing them as well. Everybody with me here? And then ultimately, the reason we generate this data is because we are genomicists. We think big, we think systematically, and we want to integrate all this data computationally to predict the genes, the regions, cell types, the circuits, the mechanisms of action. And once we have those, we've basically established a series of collaborations across every aspect of human biology to start understanding if I now manipulate mesenchymal stem cells in muscle or adipose, or neuronal co-cultures with microglia and astrocytes and vasculature and immune perturbations, then what is that function changing when I perturb the genes that we predict to be drivers? And also in mouse and animal models, how we can do that in the context of complex circuits. Is everybody with me here? Awesome. So I was actually planning to give a talk, not just give an introduction, but I'll just like fly through it. So basically, number one, we build reference epigenomes to predict what are the disease-relevant tissues. Number two, we look at how these epigenomes vary genetically across individuals and with transcription across individuals and in the context of disease. Then go into single cell resolution to understand both epigenomic and transcriptional variation across individuals. Then we look at the cell type specific genetic effects of these variations across single cell, bulk, genome-wide association studies, and quantitative trait loci that modulate expression or methylation, etc. We integrate multiple phenotypes together. We basically look at the convergence of these processes across many tissues, and then we build high throughput ways to manipulate those. So we basically built reference maps of the human epigenome. If you have a phone, you can just scan each of those and sort of look at the different papers. But basically, this was published in a journal issue with like 20 papers from our team that basically sort of painted the largest map of the human epigenome to date, allowing us to now see how genetic variants associated with different traits localized in control regions, in these regulatory elements that are active in different cell types. And that's dramatic. It basically says that if you look at genetic variants associated with height, with blood pressure, with cholesterol, with inflammatory bowel disease, you can actually see that they enrich in enhancers of different tissues. That basically allows this. So Andrew Law, he showed this figure from my paper at a cancer conference, and he says, well, Manolis, writes a bunch of papers, but even I can understand this figure. You can read this, and it says height. And then you can read that, and it says human embryonic stem cells. So height variants act in embryonic stem cells. Blood pressure variants act in the left ventricle of the heart. That's where the blood pressure is built up prior to being pumped in the rest of the body. Cholesterol variants act in the liver. And when I say act, what do I mean? We're not even looking at genes here. Genes are completely out of the picture. We're just looking at the epigenome and what regions of this packaging, these little boxes that says living room 1, kid 1, we don't have 2 living rooms, how they act in different tissues. So this is all these circuits, these control regions, these light switches. These are not the light bulbs, they're the light switches on the wall. And it gets super interesting. If you look at fasting glucose-related traits, a mark of type 2 diabetes and type 1 diabetes, you basically see that it's the pancreatic islets where insulin is actually generated. Where if you look at inflammatory bowel disease, inflammatory immune cells, great, check. Bowel disease, digestive tissues, great. If you look at Alzheimer's, well, I expect you guys to put a lot of money on the brain, if you want to guess. And that's where I would have put my money as well, but I would have lost it all. Because it turns out we saw zero enrichment for Alzheimer's and brain. That's a place where you just yell at your postdoc and say, go find that result. No, I'm kidding, you'd never do that. That's the place where you humbly say, well, maybe the books are wrong. Or maybe whole tissue of brain does not capture the subset of cell types in the brain that are the direct impact of genetic variants associated with Alzheimer's. Instead, we found that CD14 plus immune cells that mark both the circulating monocytes in your blood and the resident macrophages in every one of your tissues, including the microglia, the resident immune cells of your brain, are in fact showing a dramatic enrichment. And indeed, we went into those brains and we basically said, well, let's sort nuclei and then select neuronal cells, immune cells, and then glial cells. And what we found is that that enrichment was pointing very specifically to the immune cells that are only making up 5% to 10% of cells in your brain. So that basically says maybe we should be thinking about Alzheimer's as an immune disorder. And when we published that in 2015, like many people said, hey, I told you so, but for once we actually knew what was going on. And when all of these drugs are failing, focusing on the neurons, that's perhaps because that's a downstream effect of something that actually starts much, much earlier. So we've since expanded from 100 epigenomes to 800 epigenomes. We had another paper in Nature just a year and a half ago. Basically, it's here, looking at sort of this, incredible map. And just to be blunt, schizophrenia, neuroticism, etc., they all map to brain. It's not like our brain samples were bogus. No, it's just that excitatory neurons make up the vast majority of the brain. It's also the place where self-reported math ability, intelligence, educational attainment also localize. So I'm sorry again to be so blunt, but yes, there are genetic differences that actually impact the way that your brain works, just like every aspect of human biology. They're having minute effects, but they are immensely reliably found in the biological processes that make sense. By contrast, you see Alzheimer's localizing here in the blood. And, liver with cholesterol, filtration traits with kidney, and so on and so forth. This is also exciting because we can actually take these multifactorial, multifaceted traits and start splitting them up into their parts. Basically say, listen, coronary artery disease affects everything, but if we take the liver subset, then we find very different biological processes than the coronary artery subset, than the adipose subset, and so on and so forth. So we can actually start now mapping these genetic loci to their target genes through these correlations and start linking together for every locus in the genome across these hundreds of thousands of genetic loci, maps that we have made freely available. You can click here and browse through tens of thousands of these pictures that basically tell you how genetic variants are in fact mapping to target genes in specific tissues with specific targets and specific postulated mechanisms of action. So that's what we want to do for every aspect of human biology. And that's looking at reference maps, but we can also do that for these intermediate maps that vary from person to person, to basically look at not just a reference epigenome, but also the epigenome of this person, of every person in this room, which is actually different from each other. And we can look at how these are painting a path of causality from genetic variation to disease. And when this path is extremely large and tiny little effects, these arrows are much larger. And that's very exciting because there's only so many different variants that influence any one enhancer and so many enhancers that influence any one gene and only so many genes that influence any one endophenotype. Is everyone with me here? So basically we're trying to break up this very large path that basically leads to these tiny effects into a smaller path where we can actually find stronger effects. And if you look across 700 individuals from our cohort to look at genetic variation, epigenomic variation, and phenotypic variation, you basically see that if you tell me the genotype of this person at conception, they don't even need to be born, I can tell you what their methylation level in their brain at 93 years of age will look like. This is amazing. It basically says that genetically, for 50,000 loci, I can actually start predicting the epigenome. Why is that exciting? Because instead of doing a genome-wide association study, this very long arrow between genetics and disease, I can basically do smaller arrows. I can first look at genetics to methylation, and I can predict that with a few hundred individuals instead of tens of thousands, because these effects are stronger. I can also look at the bidirectional correlation in a small number of individuals between methylome-wide association studies, how DNA methylation co-varies with disease. No longer causality, because that's a bidirectional arrow. It could be just correlation. Disease could be changing your methylome. But if I start looking not at the observed methylation, but the imputed methylation, the genetically imputed methylation, then I can actually find causality. Because I can predict that before you're even out of the womb. And if that correlates with disease, then I have a unidirectional arrow again. Everybody with me? So this is very powerful. So we can basically do that to find now hundreds of genes associated with Alzheimer's. For every one of those, we have the cell type of action, this is neurons, the directionality, whether it's positively or negatively associated, and a bunch of new loci that were previously not visible because the genetic associations alone have this tiny effect across this very long arrow, but the moment we bridge that gap, we can actually find bigger effects. And of course, instead of doing this at the tissue level, we can now start doing this at the single cell level. So we have basically profiled more than 2,000 human brains, tens of millions of human cells across Alzheimer's, frontotemporal dementia, Lewy body dementia, ALS, Huntington's, psychosis in AD, schizophrenia, bipolar disorder, Down syndrome, autism, depression, suicide, and the list goes on. And that's just for the brain. And we've looked at multiple regions of the brain, multiple phenotypic measurements of RNA expression, of DNA accessibility, and of course, dozens of cell types, including neurons, astrocytes, oligodendrocytes, microglia, vascular cells, and the list goes on. In the first paper that we also published in Nature a couple of years ago, we basically looked at how early Alzheimer's versus late Alzheimer's versus non-Alzheimer's changes dramatically the expression of dozens of pathways and genes across dozens of cell types. We have since expanded in a paper that was just submitted a week and a half ago at how they change across multiple brain regions, how they change across dozens of different subtypes of excitatory neurons and inhibitory neurons, and how these change across different brain regions. And very importantly, we can take every single cell and start predicting the Alzheimer's-like transcriptome of that cell based on all of the annotations of the individuals, but now applying this at the single cell level. So for every cell, we can say, is it control-like or Alzheimer's-like, and how far down that path it is. And that's exciting because we can now take all of the genes that are known to be genetically associated with Alzheimer's and then see where do they change the most in expression, early, late, or middle. And therefore, when we want to intervene therapeutically, you probably don't want to go here. You want to go here and basically change the root causes of disease rather than the manifestations. And we're able to do that spatially. We're able to now visualize in tissues, in histology samples, the expression of individual cells. You can see here the arteries of, again, open heart surgery, living human hearts, where we can take these sections and basically see the expression of individual genes in individual RNA molecules in the context of their spatial organization. And we're basically using these deep learning transformers to now translate between RNA, the epigenome, imaging, and a huge diversity of phenotypes. We're learning these modules for how these genes are behaving together. We're learning how genetic variants, in fact, impact individual genes in individual tissues. We're able to classify individuals based on their transcriptome across dozens of cell types and predict schizophrenia versus non-schizophrenia individuals. And when we find exceptions, it's always interesting. This person here, this guy, has expression patterns that scream out schizophrenia, but he was not diagnosed. Instead, his son was actually diagnosed with schizophrenia. What could we do if we had these expression patterns for every person in your family? We could basically say, well, this person was not diagnosed, but genetically and transcriptionally and epigenomically, they appear to be alongside that spectrum of the disease manifestation. We can find the regulators upstream and start giving handles to therapeutic manipulation of large processes involving hundreds of genes, which are targets of these regulators. We can basically look at psychosis, not just in schizophrenia, but psychosis in the context of AD, which is actually associated with this dramatic repression of excitatory and inhibitory neurons, leading to these visions rather than overactivation as one could have naively assumed. We also looked at this single-cell dissection of two different neurodegenerative disorders, a motor disorder and a personality disorder, acting in frontal cortex versus the motor cortex. And basically, we found that, in fact, they are the same disorder, that the same cell type, these long-range projection neurons, which are known as von Economo cells in the frontal cortex and BET cells in the motor neurons, how these are actually altered and along this whole spectrum from ALS to FTD to all of these sort of both tau pathologies and TDP pathologies and A-beta pathologies, we're basically finding these dramatic signatures. So we have a series of success story after success story. Here's another Nature paper that we published just earlier this year, basically dissecting the human vasculature at single cell resolution. This paper is currently in final stages of review, looking at how that vasculature changes in the context of disease. I had actually an olfaction story that I'm going to squeeze in here, which is basically how do you get this diversity of receptors? The way that you do that is by having every neuron, every olfactory neuron, express only one allele, not even both alleles, only one allele of only one gene. How is that possible? Despite the fact that these olfactory genes are densely clustered. These are all of the olfactory receptors in one of several clusters that you see across the human genome. Epigenomics works in regions. You can repress a region or activate a region. How can you get this unique signature? And what we found in collaboration with another Greek, and in fact two Angelas, so there's Angela and Angeliki, so there's the two devils and the two angels somehow. What we basically found is this amazing signature of this repressive mark, H3K9 trimethyl, and this activating mark, H4K20 trimethyl, which is found specifically in these olfactory receptor clusters. And if you sort all genes in the genome based on that signature, what you basically see is that chemoreceptors are by far at the top of the list. So from the epigenome, we have a distinct signature which actually marks all of these chemical receptors. Again, story after story, I could just go on for a while, but I'm going to tell you one more vignette here of these hallmarks of disease. We can basically take Alzheimer's as a monolithic disease and then wonder why every clinical trial is failing. And the reason is that it's a polyfaceted disease. But what we can do using our thousands of post-mortem human brain samples is actually start looking at what are the hallmarks of disease. How can we now understand Alzheimer's in terms of its molecular basis in the neurons, in excitatory neurons, inhibitory neurons, oligodendrocytes, astrocytes, microglia, and basically recognize the distinct pathways and the distinct paintings of every patient based on those pathways. So we have these transcriptional hallmarks. From those, we can find the distinct patient subgroups, and these actually show distinct diagnosis and prognosis signatures that we can then use to prioritize treatment. So that's the vision of where we want to go. And Andreas mentioned that olfaction, of course, has a huge role in appetite, in eating and exercise, etc. And basically, this is our study showing just a week ago this balance between obesity and exercise, that there are dozens of pathways that are shifted across multiple tissues when you exercise, and dozens that are shifted when you eat high-fat diet. But in fact, the two are actually pushing against each other. And then just very briefly, we've basically developed these modular and programmable constructs that allow us to now start manipulating those. We're developing these extremely high throughput ways of measuring the impacts of those. We are able to measure 7 million enhancer elements in reporter constructs in a single experiment, enabling us to now sort of figure out high resolution marks of what are the nucleotide drivers of enhancer activity across these 500 nucleotide blocks. We can say, no, it's these 5 nucleotides that matter, and pinpoint individual regulatory motifs, individual regions of evolutionary conservation, and we're now able to disrupt those in mouse brains to basically see how they're acting. We've now partnered up with Brad Penteluta and Marika Zidnik. We have our retreat in next Tuesday. What we're basically trying to do is now start synthesizing therapeutics using deep learning methods, understanding all of these incredible wealth of information across both genomic information and also structural information to design new molecules, to synthesize them using click chemistry, using peptide synthesis, and to basically start testing them. So where do we need to go? We need to transform pharma. We need to enable personalized medicine. We need a coalition across computer science, biology, chemistry, biotech, finance, pharma, enabling patients, bringing together hospitals, and basically thinking about disease in a prognosis way, in a pathway-centric way, in a rational way. And the only way to enable personalization is get rid of this false dream of, a pill for every person. Instead, basically think about a pill for every pathway. Because a pill for every person is economically untenable. A pill for every pathway basically means that we can now leverage these sharing of pathways across individuals. And there's only a small number of these common pathways, and we can manipulate them systematically, and every patient will be a combination of pathways based on their burden. So that's where we want to go. We want to basically use deep learning, use sort of this diversity of representation learning across the human cortex, basically start understanding how that architecture that is so pervasive in olfaction is in fact pervasive across every aspect of human cognition, how in fact this encoding of order, light, abstractions, knowledge is actually different and unique in a person-specific way across every person. Understand the diversity of, all of these olfactory neurons that I mentioned earlier, every one of us carries hundreds of olfactory pseudogenes. All of these olfactory genes are constantly getting lost. Why? Because we have tricolor vision. We can tell rotting fruit just by looking at them. We don't have to smell them. And therefore, every person has a unique sense of smell based on the unique subset of chemoreceptors that each of us has, and then, of course, the unique combination. But that's an opportunity to start thinking about multisensorial engineering, to use olfaction as a window to neurodegeneration, as we heard earlier, as a model for distributed perception, as a way to reprogram metabolism, rewiring appetite and sensation with smell recording. Thinking about synesthesia and connections between the senses, between cognition and emotion. Think about a systems level understanding of the biological processes and functions, the epigenomic basis of memory formation, and this multisensorial engineering. So as we start thinking about human-machine interfaces, as we start looking at deep learning and this translation between all of these polyfactorial and polymodal ways of thinking about the world, we need to truly embrace this sort of multi-systems engineering, multidisciplinary approach that, again, our host here embodies so well, and this conference has highlighted so well. I am ridiculously fortunate to work with this unbelievable group of interdisciplinary scientists. So this is our annual retreat in 2021. And we are just thrilled to learn more about this amazing field that you guys are sort of starting to dissect and play out. And I really want to sort of say that this is really a model for human cognition and distributed perception. Every one of us is different, and that's what makes the beauty of humanity, like the joining of all of these brains from just such different perspectives is, I think, the secret to the enlightenment and the current revolution that we're undergoing now. So I'll stop there and let you guys move on. Thank you.
That was so generous. Thank you so much.
Thank you.
As I would like Shugong to come up here and ask you a question. He's your fellow geneticist. Everybody else, come to the Museum of Science where we'll have Manoli, and we can harass him there with a lot of questions. I know we all have a lot of questions. Shugong here.
I do too.
We will harass you, I promise. He needs the other one. I'll set your computer up while you ask your one and only question. Yeah, I got it. You just ask your question. I'll fix it for you.
Fantastic. I'm so delighted to see computer scientists study genetics. Without them, biologists could not entangle the complexity of science.
Well, without biologists, computer scientists would just be pushing bits around.
Computer science is a fantastic tool to solve the complex problem people look at the reductionist single cell.
That's exactly right. That's exactly right.
So you look at all great pictures, so on.
We have to
Now,
embrace systems.
Yeah.
Yeah.
So this is fantastic. Now I want to just ask one question about epigenetics. Epigenetics, you can have genetic, everybody making up the genes, but how to turn the gene on and off,
Yeah.
a lot regulated by microRNA
Yeah.
and by environment
Yeah.
and by other things. That is still one of the unsolved problems.
I completely agree. I think we have so much more to understand about epigenetics. Again, I showed you this slide where I think of epigenomics as the great integrator, where you can basically sort of capture aspects of your environment, aspects of your genetics, aspects of your disease status. They're all encoded there in the epigenome. So if we can better understand the epigenome of every single olfactory neuron, every single perception neuron, understand how that hierarchy gets formed through development, through education, through cognition, through early life exposures in every person. I think that's really sort of the next frontier for how to understand the human condition. So I couldn't agree more.
As you mentioned, the identical twins,
Yes.
Yeah.
they smell differently,
Yeah.
and develop different personalities.
Yes.
That's the
because
total
of the
epigenetic.
epigenomic programming of all of those neurons. And in fact, I'm going to go a step further and say that perhaps the way that they encode the smell of an apple or an orange is actually using different combinations of neurons, because all of these are stochastically turned on and off and they might both call it orange or citrus, but their actual neuronal encoding of that is different. And the example that I like to give is if you take GPT-4 and you want all of the knowledge of GPT-3 to be transferred to GPT-4, you can't just take the neurons and map them. That's why I had this picture of the human-machine interface. You can't just simply decode these intermediate representations. You have to actually think about the whole systems level and how they're manifesting through words or through sentences or through abstractions, etc. But I think, understanding that can. Anyway,
Thank you
so
so
much
much.
more to talk about. So you guys
I will do a very quick introduction of this
I want to
figure.
say one word. The Greek scientists now make a renaissance again.
Again. Hey, we did it once. We can do it again.
The Chinese side.
All right. Thank you so much, Manoli. And let me introduce our final speaker for the day, who's going to take us out of this wonderful experience, I think, for all of us, I hope. I can judge by the body language that everybody's enjoying it. Xu Guang is the reason I'm at MIT. The story I will tell you another time when we have more time. But he is the actual James Bond of science who crosses fields and is afraid of nothing, even though he called me that. I think he is truly fearless. He has been all over this planet doing incredibly important science. We co-founded the Molecular Frontiers Foundation with Xu Guang. We co-ran the Molecular Frontiers Inquiry Prize, the first prize for questions on the planet, which was given at Stockholm to young children for asking a good question. It was the first prize in the history of the planet given for questions. We always reward answers. Xu Guang is a curiosity fetishist, or a curiosity enthusiast, so am I. So he will talk to you about curiosity and what has driven his incredible career and achievements. And the one logistic thing I will say is that we will meet at the Museum of Science 45 minutes after Xu Guang's lectures end, so probably around 6:15 or so, because it takes only 10 minutes to get there via an Uber or car, but some people might want to walk because it's a nice walk. And I don't know what the weather's outside, but...
It's a beautiful.
Okay, so maybe it's a walk for all of us.
Better than the picture.
Okay, better than the picture. All right. So 45 minutes after the end of Xu Guang's talk, we meet at the lobby of the Boston Museum of Science. It is an unstructured, self-assembled tour. I'll be there, Manoli's, and his family will be there. I just heard my kids are not coming.
Oh.
They're coming tomorrow, though, and they will be asking questions on stage. So I'll give you Xu Guang Zhang, a true powerhouse of MIT.
Yeah. Thank you, Andreas, for inviting me to this talk. And we see so many great scientists, and you make a return of the leading scientist from Greece. How many people have seen this picture? And you're actually in this building. This IM Pei designed MIT into the building. Can you see? The M, I, and T. What is a design genius? Because after that, we designed Paris, the Paris pyramid. Okay, so we have 5 senses, actually 6 senses, including temperature. So people have won the Nobel Prize for hearing, 1961, vision, 67, smell receptor, not the actual smell. The smell receptor, 2004, and the touch, 2021, and the temperature, 2021. However, how we really smell, the chemistry part, we still do not understand. So hope Manolis and the others will crack this hard code. So here is the smell. Actually, our smell receptor not in our nose, it's on our eyebrows here. So this actually is a photo from Nobel Prize for 2004 for the winning the receptor. All the different receptors, as Manolius said, one gene expresses a single cell. About 426, the olfactory receptor in human, but one of them expresses one. Also the 500 pseudogenes. Maybe those pseudogenes are not real pseudo, because we do not understand that. However, from just assumed 400 function genes, we can smell essentially unlimited numbers. Could be billions or trillions. Okay. Here, as you can see, the human down the bottom part, it's a very small number of olfactory receptors. The largest one is elephant. People say elephant can smell water. Human cannot. Why the dogs and the mice can smell better? One is have more genes, and 2, they are much closer to the ground. Yeah. Our eye have evolved over the millennium. With standing up, can see far distant dog and mice cannot. Surprisingly, the cow and the horse have not receptor genes. It's not for their survival. Why? The question still remains unanswered. As you can see, highly evolved organisms, the monkeys, orangutan, chimpanzees, have small number of factor genes. Now, I will take to different part. And there are 2 classes proteins in human, or in all living system. One is called the water-soluble kind. It's circulating water in the blood. Hemoglobin is one of them, lysozyme, and many enzymes. The other is embedded protein in the membrane. It's called membrane proteins. In this particular case, it's a G protein coupled receptors. Smell receptors, or olfactory receptors, are the same family of G protein coupled receptors. That the receptors embed in the membrane. So it's like a window or door in a house. If you take a window or door out of the house, it becomes not functional. So this kind of protein is extremely difficult to study until recently, people use cryo-EM. And also, in protein is made the most alpha helices. As you can see, this circle, the right-handed helices, and the hemoglobin, the same kind, the alpha helices. On the left side, hemoglobin, the alpha helices is totally water soluble. That's why hemoglobin can circulate in your body. On the right side, the olfactory receptor alpha helices in the membrane are totally water insoluble. How could that be? The same alpha helices. Turn out, nature has evolved 3 kind alpha helices, and 3 different types. One is type 1, it's water soluble kind, and type 2 is water insoluble kind, it's in the membrane. Third, it's a half and a half. Okay. In those 3, the structure are absolutely identical. On the top, 1.5 angstrom per amino acid rise, and 100 degree per turn, 3.6 amino acids, 360 degree, 5.64 angstrom per helical turn. No difference. Okay. So turn out the 20 amino acids here in the image. If you look, the 20 arranged by the size. Okay, smallest to biggest. If you look in the middle, V and T in the middle, they look indistinguishable in the 1.5 angstrom resolution of amino acids. And as look on the second row, L, leucine, and aspartic acid and asparagine look almost identical. And the glutamine and glutamate also very similar to leucine. The 4 of those are water soluble, leucine is not. Likewise, the valine on the top is water insoluble, the threonine is water soluble. Finally, look down the last row is phenylalanine, it's water insoluble, and the histidine and the tyrosine are water soluble. But the shape looks more or less the same. Here explain there's several amino acids that can interact with water. The glutamine and asparagine can interact with 4 water molecules, and the serine, threonine, tyrosine can interact with 3. So that's how you make water solubility for the amino acids. Based on these 2 things, very fundamental chemistry, and people have looking over this for at least 70, 80 years, but hidden in plain sight. We didn't pay attention enough. Okay, no people in lectures and the textbook emphasize that point. So if you look, based on these 2 principles, I conceived a called QTY code for protein design. So we can now replace leucine with either asparagine or a Q is glutamine, or with valine with threonine, and phenylalanine with tyrosine. Now you have The circle is alpha helix. On the left, it's water insoluble, mostly green. So on the picture down the bottom, the yellow part is mostly water insoluble and hydrophobic surface. So now we can change all the QTY, change all the leucine valine isoleucine phenolene, change QTY, now become water soluble version. Now you can see this yellow part is reduced substantially. Here is two examples of the GPCRs. So because olfactory receptor is all GPCR, so we study GPCR in the similar family. So this 6L4 on the left side, you can see the green part and the cyan color, which is like water color, is superimposed very well. It's by AlphaFold 2, by the way. Yeah. Thanks AlphaFold 2, it came out July 2021. Completely transformed science. That's why they deserve the breakthrough prize, for sure. And if you look at the top, look at the, essentially they are the same. The difference is it's less than 1 angstrom. On the right one is another GPCR, the same way. Amazing. So how it changed it about 20%, in some cases 30% amino acid in the protein, and the structure, overall structure did not change, and function did not change. We already published papers, we don't have to go into detail. So the magenta color is the crystal structure of the 6L4. The green part is predicted by AlphaFold 2, 6L4. The cyan color is the QTY version, the water-soluble version of the 6L4. So here it's labeled hydrophobic. Of the native one has more yellow color, and the QTY variant has less. Not only the QTY could apply to GPCR, but to other membrane proteins. In this case, it's called glucose transporters. And glucose transporter, the magenta color is crystal structure, a cyan color is QTY version of the protein. Now the protein can be produced in large quantities, can be studied, and can be used for biotechnology applications. The two different glucose transporters. Now, I'll show you here. And everybody in the lab use the glove for working in the laboratory. Here I call hydrophobic. Yeah? Hydrophobic is a surgeon used for operating you. And for semiconductor people working in the companies have to use this to handling all the chips. The function with the glove and then without are no difference, identical. So on the left is hydrophobic, on the right is hydrophilic. That I explained the alpha helices can be changed. Now, in collaboration with my colleague at Uwe Schleiter in Austria, in Vienna, now he discovered a class of protein called acylaire protein on the two-dimensional crystal on the cell surface. This is on the cell surface. Here is imaging on the silicon wafer. Because that two-dimensional crystal lattices, now we can also make a recombinant protein, make an individual point here on the color white part as a linking point for another protein. That's exactly what we did. We can now linking those proteins down the bottom part here, making G protein coupled receptors become water-soluble version, QTY variant, and then now can put a linker onto it, like FC domain of IgG. And now this acylaire protein is also has protein A, protein G, and the recombinant protein binding to it. Now, down the bottom part here, you can make a recombinant protein two parts that never meet in nature. One is eukaryotic, for example, G protein coupled receptors. The other acylaire protein is bacteria, here especially. So now it can form the same kind of protein. Now you can anchor those onto silicon chip, onto silicon wafer here. And not only can anchor, the density is about 4 trillions per square centimeter, extremely high for that. It's all nanometer because the 2-dimensional crystal, they can arrange this. You don't have to worry about that. Yeah. So now you have extremely high density. You can use now, make a device as a sensor in collaboration with people in physics department here. And essentially make it into a sensor, and it can measure this sensitivity of measure ligand binding without other fancy instrument, SPR and UV spectroscopy, or many other fancy ones. It's measurements by electricity. So electrical readout, and very sensitive and quite specific. This paper is now under review in journal. So you can see one of them, and the green part is one negative control. Down the bottom have nothing bind. On the top part is correct ligand and binds to generate another signal. So down the bottom part with 6L5, there's no signal can be detected. Well, Leonardo da Vinci it's a great master science and art. Yeah, he said, simplicity is ultimate sophistication. So, Maki, let's talk about the DNA. Yeah, DNA is like a code, just like a computer code. You're right, but one was the simplest code. Yeah, once you read out one part, you know immediate other part without making any mistake. If you make a mistake, it's a mutation, as Makita talked about over and over. So that's why DNA is one of the simplest nature-made code. So now I call the code, the DNA code, A with T, G with C. Now I also consider my QTY code a simple code. L with Q, V and I with T, and F with Y. It's bidirectional. It's so important, it's bidirectional, like DNA. Because of bidirectionality, we now have another paper on the review. We changed the water-soluble protein. It's a human cerealbumin, very soluble protein. Yeah, it's circulating in your body. And made that partially water insoluble, and that protein self-assemble into precisely nanoparticle that can use for drug release. And very good. That paper is on review. I didn't put the slides on it. So, Andrea's dream. We have hearing now. We have phone. We can hear. Our phone has touch. Our phone has vision. We don't have smell and taste. Since 80% taste is actual smell, you only need to make a smell. That has been Andrea's dream for now more than 10 years, 15 years. Want to make a phone that can do smell. So Andreas has been working on this very hard. Hopefully, eventually, will succeed. Here is the founders of modern molecular biologists. My mentor, Alexander Rich, with Francis Crick, Jim Watson, and Leslie Ogle in this house, the Crick's house in Cambridge, with a yellow right-handed the helix. It's the alpha helix. That's before Crick discovered DNA double helix. This was installed 1951. Crick discovered double helix 1953. So Crick said one could not be dedicated to anything unless one believed in it passionately. We saw how passionate Marquinhos was about use AI to study genetics. And Andreas very passionate about generate design a new phone that can smell. So we do need this passion and to do something dedicatedly. All right, thank you very much.
Thank you very much. Questions for Shubham, who finished on time? Wonderful. Questions for Shubham? You actually answered my question. I was going to ask, did you do it the other way? Did you make it from hydrophilic to hydrophobic? You have a question?
We did.
Right. Give him a mic.
Yeah.
Thank you so much for the beautiful lecture. So,
Hold it for the web so they can hear you.
So my question is going to be a very simple comprehension question. So basically, you're talking about this QTY code and this complementarity, and this involves only 6/7 amino acids.
Yes.
My question is, what about all of the other ones? Are there still parts of the code? Because if you think about the expressivity, I mean, again, part of the reason why proteins were initially thought to be the unit of inheritance is because DNA seems so complex. There's so much expressivity with the 20 amino acids than with the 4 nucleotides. But if you look at all of the other amino acids, I mean, in a way, there must be many more interfaces, and there's just so much more room. And as an engineer, I can see the desire to great, you now have a simple code, let's use that and design machines that are just very simple, and that's what Leonardo da Vinci would do. My question is, nature is sometimes very messy, and it finds ways of doing things many, many different ways. Could you comment a little bit about what fraction of this interface code is QTY based versus everything else, and are we perhaps missing many additional such codes that we should go out to discover? Now that we have AlphaFold, we can start systematically looking for such interfaces.
Well, nature only gave us 20 amino acids. With my image. Can you put back? Nature only gave us 20 amino acids. If we can put the slides back on, it would be very helpful. So far, QTY change is from change hydrophobic to hydrophilic, and vice versa.
Yes. So one interface water.
Yes. There's only one more. It's between serine and cysteine on the third one and the fourth. Yeah. And also phenolene with histidine. Yeah. And all others look at very different shape. So it's very difficult to change. If you change tryptophan with glycine, you sure make a mess. And if you change leucine with tryptophan, you also make a mess. That's so different shape. And so this is important. We cannot do what we want in nature. And nature gave us this code. We have to use this as a starting point.
As a follow-up, basically, as an epigenomicist, I can't help but think about all of the post-translational modifications of these amino acids.
Yes.
I'm curious how much more versatility you get.
It's actually the only 3 amino acids, or 4, that's involved in post-translation modifications. It's serine, threonine, and tyrosine and asparagine. Only 4. Either phosphorylation or glycosylation. And there are no modification for tryptophan, arginine, and also actually lysine can also get acetylated in the histones. So there are not really many, and you can do modifications. And most, especially hydrophobic residues, they have no reaction motif for that. So there are 5%, you can do things. However, change 1-to-1, then you need to follow this, what the nature gave us, a playbox. Yeah.
Dimitri, you have a question?
I do.
Hold on, let me give you the mic. Go ahead. I think this might be the last question now of the day, because we have to go.
In my law review article, I cite your work, and I wrote that in 2009 is when proteins, you were able to create olfactory cells in the lab through proteins.
Yeah.
How did you get to that stage, and what exactly did that mean in leading Andreas to kind of get into the olfactory world?
Well, there's a book Andreas and I read and called Emperor Scent. and described one scientist who was obsessed with smell, Luca Turing. And that was a book, stimulus to study olfactory receptor. At the time, I was thinking to change my research directions. I worked for 15 years on the called peptide self-assembly hydrogel material science and form a company. Company became successful under MIT's rule. Once you're successful at something, you do something else. So that's what I did.
Got it. Thank you.
All right, I think,
All right.
unless there's other questions from the audience?
Can I do one more question?
Okay, last question from Ilya, and only because you're from Aromics. But you need the mic.
For a wonderful talk. I just had a mechanistic question. So what actually happens when you take this modified...
For the web, you need to do this, sorry.
So what happens when you take the modified DNA, you get your, let's say, hex cell to express it, does the protein actually still go to the surface on the cell, or does it stay solubilized? I'm just wondering, like, what does the ER do with the protein?
Good question. And indeed, that's the question we answered. And when you change small amount, like say 7-8%, the protein that's receptor modified QTY versions can still go into membrane. But at a reduced the efficiency, in order to activate that protein, G protein coupled receptors, you need much higher ligand concentration to activate. But if you change all of them, like what we did later, and the protein can no longer go into membrane anymore. Yeah, because that's exactly what we wanted, to make olfactory receptors to put onto the chip. And I just sent the protein sequences to Andreas, and so they're going to actually make the receptor to try it out.
Thank you.
Okay,
Yeah.
this is it. Thank you so very much. We will see you at 6:15 in the Boston Museum of Science. And tomorrow
Okay.
we're starting here at 9 a.m. Thank you.